Good day, grade 12 learners. Welcome to today's Life Sciences lesson. My name is Sina Sekheche, and this lesson is brought to you by the Free State Department of Education in collaboration with Pagama Research and Development. So, today we are going to be focusing on the revision for paper 2 that you will be writing on Monday, the 3rd of June. So, we need to check what are the topics that we will be discussing for this. Right. So the content that you need to cover for Monday's paper 2 are the DNA, code of life, meiosis, and genetics and inheritance. On your right there, it shows you how many marks are going to be allocated for each topic. So for DNA, code of life, you are going to expect 42 marks out of the entire paper from the various questions that you will be getting. And then for meiosis, you are going to write out of 32. For genetics and inheritance, you are going to write out of 76. So in total, you are going to be writing a paper out of 150. So you have already written your paper 1. I hope it went well. You were able to see the structure of the question paper, and now you are familiar with the paper. Right. So just to check, you are writing your paper 2 on the 3rd of June. It is going to be out of 150 for 2.5 hours, meaning 1 minute per 1 mark. So your question paper is going to be structured into 2 sections. You are going to find section A and section B. Your section A is going to consist of short answer questions, objective questions or your multiple choice questions, and your terminology, which is very important in life sciences, and the matching type or the matching column. All in all, for section A is going to be out of 50. And then your section B is going to have a variety of questions that are divided into 2 questions of 40 marks each. And those 40 marks can further be divided into 3 to 4 sub-questions. So you know your 2.1, 2.2, and 2.3. And then question 3, 3.1, 3.2, and 3.3. So that is your paper, the structure of how your paper is going to look like. And then there are a few things that I would like to highlight, which causes you to lose marks when you are writing. So these things are written on the marking guidelines. These are the principles that we use when we mark your papers. So please take note so that you are able to answer your questions very clearly. The first principle of marking is that if there are 3 reasons are required and you give 5, let's say you are feeling clever, you want to give us 5. The principle says we must mark only the first 3, irrespective if the rest of them are correct or incorrect. So if you've started with the incorrect ones and the correct ones are number 4 and number 5, you are only going to get 1 mark because we are going to mark only the first 3. So take note of that principle. Only write the required number. Right. Another principle of marking is that if tabulation is required, but paragraphs are given, candidates will lose marks for not tabulating. You know, they will say, tabulate the differences between DNA and RNA. And then you decide to write that in a paragraph. You are going to lose that 1 mark for drawing a table. And remember, a table is something that has lines for the columns and lines for the rows. So it must not just be 1 line in the middle. There should be lines between the rows and lines for the 2 columns. Right, and then you're going to get 1 compulsory mark for drawing that table. Then, if flow diagrams are given instead of a description, marks will be lost. Sometimes they would say, please describe the process of protein synthesis. And then you decide not to describe it, you decide to draw. You are going to lose marks. Please make sure that you follow the instructions correctly so that you do not lose marks for free. Right. This one is very important also. Read your instructions carefully. It says, if only the letter is asked for, but only the name is given, and vice versa, the candidate will lose marks. So you need to check if they say you must give the letter and the name. Let's say letter is C and then the name is DNA or something. So these, both of them needs to be there. Please make sure that you take into cognizance. Right. So what possible questions can you expect from the DNA topic? So you can expect terminology questions, which is a skill that you need to learn in life sciences. By now, you know life sciences has a lot of terminology. Each topic comes with its own bunch of terms that you need to study. I hope you are compiling your own glossary at the back of your book where you have all the terms defined, or you are also studying your terminology tests. So you have to know there could be a question that is asking you about terminology, interpretation of diagrams. They can give you diagrams on protein synthesis, a diagram on DNA replication, any diagram. Then you must be able to tabulate those differences. They can say tabulate the differences between RNA and DNA. Then there could also be a description where you are describing the whole process. So you can be asked this question, and you can also get a question where you have to calculate. When you are doing protein synthesis, you can calculate the amount of of nitrogenous bases, the percentage is there. So you can expect any question of that sort. And then for meiosis, same, you can expect terminology, diagram interpretation, description of crossing over, tabulation and differences between meiosis 1 and meiosis 2, the differences between meiosis and mitosis. So those are the things that you can expect from the topic meiosis. Then the last topic that you're going to do is genetics and inheritance. It is quite a big one. I think you saw that on your paper. This is going to amount to 76 marks. So these are not the marks that you can just lose. You need to know all the concepts, meaning all the terminology that you're going to use for this topic. And the terminology here, I know it can be very tricky. But if you are having a challenge with a particular term, try to use it in a sentence or make an example for it so you can understand where that terminology is used. Because with genetics, the terms are practical. You need to be able to explain in practical what that term is, where that term is applicable. Then you need to be able to do your monohybrid crosses. Then you're doing a genetic problems involving one characteristic. Remember, mono means one. So your monohybrid characteristic will be that one where you are tracing only one characteristic. Can be height, eye color, shape of the fruit, only one characteristic. So those are monohybrid crosses. And then you know there are different types of inheritances that you're going to learn also under the monohybrid crosses. We will deal with that when we come to that. And also sex-linked inheritance. Here you must be able to do a genetic problems involving two sex-linked disorders that we have studied this year. And those are hemophilia and color blindness. I know most of you are making a mistake when you are doing a genetic cross. We will talk about it. You think everything is sex-linked. You use the X and the Y. We are going to deal with that program today. Then you need to be able to know the pedigree diagrams or the family trees, which are used to study the inheritance of characteristics in a family over a number of generations. So you are going to check the lineages, or you are going to check the inheritance, where this particular disease comes from. So you remember the pedigree diagram that looks like a family tree. So you must be able to interpret that one. And then genetic engineering, the stem cell research, genetically modified organisms, and cloning. So here you must have received activities that are extract based. You have information that you have to study and answer on this topic, right? And then paternity testing, the use of blood grouping and DNA profiles for paternity testing. So how do we use blood groups to prove paternity, and how do we also use DNA profiles to prove paternity? I hope you do know those. Right, so this is just the scope. So now we are starting with the first topic. Remember, this is a revision. This is to highlight those important things that you might miss when you are studying. You still need to go study and get all your question papers and answer the question papers after this session while this information is very fresh in your mind. So we will start with DNA and RNA. Just to recap on the structure, you remember the structure, the location, and the functions of DNA and RNA. So if you can just recall where you are sitting, you are checking yourself. Do you know how the structure of DNA is? We say DNA is double helix. What does it consist of? It consists of nucleotides. What are those nucleotides made of? You have a nitrogenous base, you have the phosphate group, and you have the sugar group. So you must know what type of sugar do I have in DNA. You have deoxyribose sugar. What type of sugar do I have in RNA? Ribose sugar. So you must know, where do you find this DNA? DNA, you can find it inside the nucleus on the chromosomes, and then RNA you can find where? You can find mRNA leaving the nucleus, you can find it on the ribosome. So you check yourself, you revise in your head that, do you know the location? So they can give you a question where they ask you, where do you find this type of DNA? Where is it located, or how is the structure? You must remember the structure for DNA. We do not speak of double-stranded only. We do not speak of spiral, but the correct and allowed term that you need to use when you are describing the shape of DNA is double helix. So DNA, you must also know that it forms genes which carry hereditary information. So that is one function of DNA. You know, you become what you are because of the DNA. So DNA is the one that carries those hereditary characteristics. Another thing, it carries or it contains the coded information for protein synthesis. Those are the two main functions of DNA: hereditary characteristics and carries the code for protein synthesis. Remember, synthesis is to manufacture or to make. So it carries the code to make the proteins. Right. RNA plays a role in protein synthesis. Then you remember mRNA, tRNA, what role does it play in protein synthesis? So that is the function of RNA. At this stage, you are getting warmer. Your mind is now starting to think, okay, so I know this content, I am comfortable with this content. Then you come to the most important, this one. There is no way your question paper is not going to have this. Remember the process, you need to be able to describe the process. So DNA replication and its significance. Why does DNA need to replicate? So to replicate is to make copies. Remember. So why does DNA need to make copies of itself? So DNA makes copies of itself so that it can be shared between the resulting daughter cells during cell division. So it makes it to have enough genetic material that can be shared during cell division. So you need to know now the process of DNA replication. How does it happen? Can you remember? You start by doing what? You start talking about the double strand and zipping and winding and what. But I know you mix the processes as you go. It needs to have a logical flow. We will look at that process right now. Then DNA profiling, you must also know. Remember the term here, we no longer use DNA fingerprinting. The correct term that we use is DNA profiling. So what is a DNA profile? We use it the interpretation of DNA profiles. So you must be able to interpret those DNA profiles when you are given. So you think of those questions where you are given a DNA profile, are you able to interpret? Use a ruler. All the time when you are interpreting a DNA profile, use a ruler. Because sometimes your eyes can deceive you. You might think that the DNA of a baby is matching a particular parent or a particular father, and you find that no, your eyes, maybe your paper has turned or something. Please use a ruler or a pen, but you must not enter the exam room without a ruler. So use a ruler. Then each person has a unique DNA profile. So that is why you are able to use it. Remember the uses of DNA profiling. It is because we want to match, because your DNA profile cannot match the DNA profile of another person unless you are genetically identical twins. A profile is represented as an arrangement of black bars. So you already know what I'm talking about. Representing the DNA fragments of a person. So you must be able to know that and know the uses. What are further uses of DNA profiling? So what do we use it for? For solving crime. You know all those reasons. Then the main one that you also make huge mistakes on is you must know the process of protein synthesis. What is this process? It is a process by which proteins are made in each cell of an organism to form enzymes, hormones, and new structures for the cells. Remember when you were in grade 10, you used to say proteins, hormones are proteins. What? So these proteins are formed by using the code that is carried in the DNA. You must be able to describe the whole process from where it starts to where it finishes. It starts with transcription. The code needs to be encoded and carried. Then when that code is carried to the ribosome, that is when it's going to be interpreted to form amino acids that are going to form a long chain of proteins. So you must be able to know how do we form proteins. So you must know that process of protein synthesis. Then the process involves transcription. Remember we said it's the first stage. So where DNA forms mRNA. Now that mRNA has the coded message for the protein synthesis. And then the next stage is going to be translation. Where what is going to happen? Translation is whereby the tRNA is going to read the code from the mRNA and form amino acids which are going to link by peptide bonds and form a protein. So this is just on the surface. And your heart must be beating now to say, do I know this process? So let us take a look at them in detail. So this is the DNA and RNA. You remember we said DNA has the sugar, deoxyribose sugar. If you find that the word or the name is too long, try to cut it in the middle somewhere so you can know. So deoxy, you can remember that. Ribose sugar. So this is the type of sugar that you find in DNA, and then in RNA you find ribose sugar. DNA is double-stranded. You see this double strand, this. But remember, we don't use that to describe the shape. This is just to tell you about the structure. So how is it structured? It is structured, it has double strands, but those double strands, we know they are helical. Right. So it is a double helix structure, and then you have a single strand for RNA. So RNA is just one single strand. And then DNA is a long molecule, and RNA is very short. RNA, you saw, sometimes it goes with just 3 anticodons or 3 codons. Very short molecule. But DNA is a very long molecule. And then the building blocks are nucleotides. So let's talk about these nucleotides. So these nucleotides are here. So this whole part here, the one that I'm circling, is a whole nucleotide for DNA. So what is a nucleotide? Nucleotide consists of nitrogenous base, the sugar group, and the phosphate group. So how do you know which one is the sugar, which one is the phosphate? The sugar one will always have this type of a shape, the one that looks like a house. So you will say that one that looks like a house is a sugar group. So that is a pentagon, hopefully, yes. And then the nitrogenous bases are the ones that are joining the ones on the other side. You have 4 different types of nitrogenous bases. You have adenine, thymine, guanine, and cytosine. So these are the nitrogenous bases that you find in DNA. Right, and then your phosphate group will always be the one that is represented by a circle or by a dot. All of these 3 things, 1, 2, 3, they form a nitrogenous, they form a nucleotide, not a nitrogenous base, they form a nucleotide. And then for RNA, what are the nucleotides? Almost the same. So you come here, what I'm circling is a RNA nucleotide. So what does the RNA nucleotide consist of? It also consists of the phosphate group It consists of the sugar group, but the sugar here is ribose sugar. And then the nitrogenous bases, you find adenine, uracil, guanine, and cytosine. So instead of thymine, now this one is instead of T. So instead of T, instead of T, you find U instead of T. So this is in RNA. So I hope you remember the structure and the molecule, how they are. So this is just to explain further what a nucleotide looks like. Remember, you have your pentose sugar, your phosphate group, your nitrogenous base. And we did say that the nitrogenous bases may differ in DNA or in RNA. And in DNA, the nitrogenous bases work in complementary base pairs. In DNA, they are complementary. What do we mean by that? We mean if a nitrogenous base is coming from this side and it is A, coming from this side it is T, this side is G, this side is C. We know that DNA is double-stranded, meaning there should be some nitrogenous bases coming also from the other strand. But these nitrogenous bases have to be complementary to the nitrogenous bases that you find on the first strand. So they form what we call is base pairing. They are complementary or they form pairing. Base pairing that are complementary. So A pairs with T. T pairs with A. G pairs with C. C pairs with G. So these are always the case. So you must remember that they are like this in DNA, but in RNA, remember it's a single strand, they are just like that. But however, when the tRNA comes or the mRNA comes, they need to be complementary. And that is where instead of thymine, you are going to use uracil. So where A pairs with T in DNA, then in RNA, A is going to pair with what? With U. Right. So I hope you remember all of this together with the nitrogenous bases, how many we have, and the structure of DNA and RNA. Then we come into the next process that you need to be able to define, which is the process of DNA replication. Remember, we said this is the process where DNA is making copies of itself. So how does that happen? Remember the steps. The first one, the double helix unwinds. Use your hands. If you feel like you've forgotten, use your hands. Sometimes you may not have this diagram here that you have here that will be showing you. But remember, you are going to say this double helix was winded up, but before it makes copies of itself, the first thing that it must do, it must unwind, because you can't make copies of itself when you are winded up. So you must unwind. Then you are going to get a mark for saying that. The second thing that you must say is that the weak hydrogen bonds between the nitrogenous bases are going to break and the two DNA strands unzip. So when you talk about the hydrogen bonds breaking, it's a mark, or when you talk about it unzipping. So you can't get 2 marks for that one. So it's only going to be 1 mark when you say the weak hydrogen bonds. So the weak hydrogen bonds are here between the nitrogenous bases. After unwinding, the weak hydrogen bonds are going to break. And when they break, what happens to the molecule? The molecule is going to unzip. So we mark it as 1 mark. So if you wrote unzip, 1 mark. If you wrote the weak hydrogen bonds are going to break, you're going to get only 1 mark. Then 3. When it unzips, now you have what? Each original DNA strand is going to serve as a template on which its component is built. So you have one strand unzipping, one strand. Now these are open. So they are open for what? For new strand to form here in the between. Okay, let's say you have this old strand, this old strand. They are open like this. So when they are open like this, they open a chance to have a new strand forming here and a new strand forming here. This is what we mean when they say they act as a template. Right. Free nucleotides build a DNA strand onto each of the original 2 DNA strands. So what is going to build this new strand? You are going to use the nucleotides that are floating in the nucleus, in the nucleoplasm. So you have your A's, your T's, your G's, your sugars, you have your everything is floating. So when they see that here is a T that is open, here there's a A that is open, here there's a G, here there's a C, the complementary ones from the nucleotides are going to come and complement here. So the sugars will come and form a backbone, and then the nitrogenous bases will complement the ones that are already open. So it will come here. A that is floating will come here. T that is floating will come here. C that is floating will come here. G that is floating will come here. So this is what is meant by free nucleotides build DNA strand onto the original strand because they are complementary, attaching to their complementary ones. So this is what we've just explained here. Then after that, this is an example to say A pairs to T and C pairs to G. We've just done that here. Right. Then this will result in 2 identical DNA molecules. So the same thing that is happening in here is going to happen here, because remember, originally here you had the nitrogenous bases that were complementary to the original strand, to this strand over here. But now they are going to be complementary to the new ones. But when you compare this strand and this one, they are identical. So when they are identical, it means they have now replicated. So that is the problem. So each molecule consists of one original strand and a new strand. One original and a new strand, the one that we drew with the dotted lines. I hope you understand this process. You are able to follow this process, and you will be able to explain this process when you are asked to do so. Then another process that you need to know is protein synthesis. How does cells manufacture proteins, or how does DNA manufacture proteins? So it's a whole process that starts from transcription. So transcription happens where? In the nucleus. So you see here, this diagram over here, I'm just going to draw it here. This diagram is your nucleus. So this is what is happening inside the nucleus. So transcription is happening in the nucleus. What is happening in transcription? You must follow. The process of transcription is almost similar as the one of DNA replication. However, there's a slight difference. So you must check out or be on the lookout for that difference. So what is happening here in the nucleus? The double helix is going to unwind. Remember, we had this also in the previous stage. So you're going to get a mark for that, yes, because we need to get the copies of those DNAs. The mRNA needs to copy the code from DNA. So it can't copy the code when the DNA is helical or it is double helix. So it needs to unwind first. So it is going to unwind. After unwinding, the weak hydrogen bonds are going to break, or the 2 DNA strands are going to unzip. Remember, we said it's already 1 mark. Once it's unzipped, it's open. And once it's open, you must now think, what's going to happen once it is open? A new strand must follow, must be copied. Right, so let's check what happens. One strand, this is very important. This is what makes protein synthesis transcription to be different from DNA replication. Only one strand, not 2 strands. One strand acts as a template. Remember, with DNA replication, we said 2 strands act as a template where new DNA is going to be formed. But for transcription, only one. Only one strand is going to act as a template for the construction of mRNA in the nucleus. So it is going to act as a strand to form construction. Don't let this word scare you. So this is to form. It's going to form mRNA. So you are asking yourself, how is it going to form mRNA? Because we have a strand that is open. Now we want that strand to form mRNA. That strand, when it's open, remember it has, let's draw it here. It is open like this. It has its own original nitrogenous bases. So I will just write NBs here. Nitrogenous bases. Now, it is going to use the free RNA nucleotides. Now you don't say the free DNA nucleotides. This is another thing that makes it different. The free floating ones in this case, in this step, are the RNA nucleotides. So you're going to use the free RNA nucleotides from the nucleoplasm to form mRNA. So mRNA is going to be formed based on what? Okay, this is my mRNA, the dotted one. This is just to take you through what is happening. mRNA, this nitrogenous base is going to be complementary to this one. This is going to be complementary to this one. This is going to be complementary to this one. This is going to be complementary to this one. Now, these ones, they have now done what? They have copied the code. They have now copied the code from the DNA strand. Now, they are now called what? mRNA. This new strand. So mRNA complements the DNA in the nucleus. So you see, it copies the code by complementing the DNA. This and this. This one and this one. This one and this one. So they are complementing. In that way, when it's complementing, it is copying the code. Then mRNA is formed. This one, you call it an mRNA. So that is how the process of transcription takes place. But once this mRNA is formed, so you see here, this is an example of an mRNA that is formed. Look where it is written mRNA, mRNA there at the top, mRNA where I am. This is the new mRNA that was formed from this DNA. You see? This DNA, so this mRNA copied the code from this DNA. It formed this mRNA. Now, this mRNA must not stay in the nucleus. It must leave the nucleus through the nucleopore. This is the nucleopore. Attach itself onto the ribosome. So you are just finishing up the step to say that mRNA that we just formed is not going to stay in the nucleus. It is going to leave the nucleus and attach itself to the ribosome. So this here is a ribosome. This structure here that looks like a potato, it's a ribosome. Right. So what is going to happen once this mRNA attaches itself on the ribosome? The process is not yet finished. The next process must take place, which is translation. What is happening during this translation? The coded mRNA leaves the nucleus through the nucleopore to the ribosome. We've already discussed this one. It attaches itself there. Then tRNA is going to pick up amino acids and complement mRNA in the ribosome. I know this step, you might not understand what is happening. So, because you have the triplet bases, so let's start with the first ones. Here you have UCA. UCA is on the mRNA. Remember, this is your original mRNA that is coming from the nucleus, this one. So UCA, tRNA is going to bring about the required anticodons. Oh, let's also discuss that. The triplets that you find on the mRNA are called codons. The triplets on mRNA are called codons. So what is going to happen? tRNA is going to bring the required amino acids. Amino acids stays there on top of the tRNA. But the tRNA must bring the complementary anticodons that are going to complement the codons on mRNA, and then they are going to release the required amino acid. And these amino acids, as they are released more and more and more, they form a chain, and this chain is joined together by a peptide bond, and it is now going to form what? A protein. So don't get lost. It is the tRNA bringing the complementary anticodons to the mRNA and releasing or bring about the required amino acid. Then these amino acids are going to be joined together by peptide bonds to form a protein. As simple as that. So please make sure that you answer as many questions on this question, then it will also make sense to you. Then DNA profiling. What is DNA profiling? This is a technique that we use to identify the sequence of bases. So what are these sequences? What do they do? The nucleotides are separated from each other in order that they are found in a strand of DNA. So they are going to separate your nucleotides in that order in which they are found in your DNA. So no two persons' DNA are the same because the arrangement of the nucleotides are not the same. So they are going to arrange them in that order when they are doing your DNA profiling. Now, nucleotides are going to appear what? As dark bands. So when you look at this diagram here, you see these dark bands. So these dark bands here, they represent the nucleotides, the arrangement of the nucleotides in your DNA. And DNA profiling is different from DNA fingerprints. DNA fingerprints is another process, but we are dealing with a profile that has your whole information. And a DNA profiling, we know we can use DNA profiling in so many ways. I would like for you to sit there right now and think of the ways that we use DNA profiling, because it's another question that can come up in the exam to say, what other ways can we use DNA profiling? If you look at this one is the crime scene. It is the sample that is found in the crime scene. And then you see there are suspects there. They are going to compare the DNA profiles of these suspects together with the evidence that was found from the crime scene. Now, if one of these suspects matches this, it means it places them at the crime scene and they have used it to solve crime. And then a question can come and say, what other ways can we use DNA profiles? Then you know you have those in your exam guidelines. Please make sure that you go check. So these are the key things that I would like for you to remember, because we cannot talk about everything all at once. We are just revising. So remember that you need to know the history on the discovery of the DNA by Watson and Crick and Franklin and Wilkins. So remember that history of what happened to the DNA fragments, who came up with the molecule, and then who stole what, who won the Nobel Peace Prize. Remember all that history of how we came about knowing that the DNA is a double helix molecule. Know the structural differences between DNA and RNA. Once you know those, you are confident about your topic. Then you must also know the differences between DNA, mRNA, and tRNA. So you must know, when do we talk about mRNA? When do we talk about tRNA? When do we talk about DNA? Especially for your process of protein synthesis. Then be able to differentiate between DNA replication and protein synthesis. I have just shown you now when we were talking about those processes. Be able to know what is transcription. And transcription is the first process that happens in protein synthesis. And translation is the one that you do later. So I like to use the sound translation to say translation happens later, because it comes after. Because some of you switches these two processes around to say, if they ask you, name the process that is happening here, and then you will say transcription, having forgotten that which one is the one that starts first. So now we're going to learn that transcription is the one that starts first, and translation, it happens later. It is the one that happens later, right? That is where now the protein is almost formed. Then DNA profiling, you must know how it is important and how do we apply it in those questions. Paternity testing, which involves both parents and the child. You must also be able to tell if they've given you the profile of a mother, of a father, and of the child to say which one is the parent of this particular child, especially the father, comparing the DNA of the child, the mother, and the father. And also the uses and interpretation of DNA profiling. Go test yourself with as many questions as you can on this topic. So we're gonna take a break for you to think about this of about 2 minutes, because then we're gonna start recapping on the topics of meiosis and check what are the key things that you need to remember before you enter your exam. So after this 2 minutes, we will start with the topic meiosis.
I want you to ask yourself these 3 questions. Number 1, what do I want? Do I want to become an A student, or do I want to become an average student? Number 2, I want you to ask yourself, how do you plan on getting there? Are you going to put more extra hours, or are you going to just sleep? Number 3, who will be with you in this journey? Because it is important that we associate ourselves with students who have the same vision as us, who want to become a student, who want to put in the extra work, who want to become better at what they do. Unsuccessful people do not plan, they just let life happen to them. But successful people are planning on how they want their lives to be. And right now, you plan on how you want your results at the end of the year to be like.
Excellence. Excellence is a habit. Excellence is a quality you have, a quality I have. It's a quality we have when we honor what is true to us. Yes, we are gifted differently. uNkulunkulu akasinganga ngokufana. Excellence is striving for your best. Whenever you have a particular project, test, homework, uzibuze, ask yourself, is this my best? Can I put in more effort? Excellence is striving to be in the top 1% of the area in which you are gifted. And so I challenge you, in every single thing that you do, in every single area that you're.
Right. The next topic that you are going to look at is meiosis. Remember, meiosis also forms part of your paper 2. You need to revise some of the topics here in paper 2. I mean, on the topic meiosis for paper 2. So what do you need to know for meiosis? Is the type of cell division where a diploid cell undergoes cell division, or 2 cell divisions to form 4. So this 2 is very important here. 2 cell divisions, because we have meiosis 1 and meiosis 2. You have meiosis 1, and meiosis 2. So those are the 2 divisions that we are talking about. But they result in what? 4 haploid cells, which we call gametes. Gametes are our sex cells. So meiosis, we form sex cells. Unlike mitosis that we did in grade 10, where we were forming somatic cells. We were forming ordinary body cells, which are called somatic cells. But here we are forming gametes. Gametes, which are your sex cells. Right. So before meiosis begins, there is going to be DNA replications. You must know that is happening. Then you must know what is happening in the first meiotic division. Then you must know what is happening in the second meiotic division. And you must know, why is it important? Why is meiosis important? It is important because crossing over brings about genetic variation. And it also ensures that sex cells have a haploid number of chromosomes. We only want 23 chromosomes. So for meiosis to happen, it is the halving effect that is making sure that chromosomes are halved, or the gametes that are going to form have half. Because if we do not have 23 chromosomes forming, you are going to have a parent who is who has a gamete that has 46 chromosomes, and another parent having 46 chromosomes, and when they come together during fertilization, now this zygote, this organism that is going to be formed, is not going to have 46 plus 46. It must have 23 plus 23 to maintain that 46 number. So meiosis plays a role in halving the number of chromosomes to ensure that gametes have a haploid number of chromosomes, which is 23, not a diploid one. And then also it brings about genetic variation. And if it was not for this, all of you and your siblings at home, your brothers, your sisters, and yes, you were going to be one person. So, but because you were born from the same parent, and those parents, their gametes have the different, they have variation. That is why the offspring, which is you, are now different people. Otherwise, if it was not for meiosis, we would have a problem of reproducing the same thing over and over. But meiosis brings about that genetic variation. So we will talk about that later. So let us get into the terminology. Like we've said previously, that all these topics in life sciences, they have deep terminology that you need to understand. So if you do not understand this terminology, you render yourself liable to failing some of the easiest questions. You know, on your question paper, question number 1.2 is terminology. You must ensure that you know your terms. So let us take a look at some of the terminology that you use when you are dealing with the topic of meiosis. Make sure that you know these terms by tomorrow. Homologous chromosomes. What do they mean when they talk about homologous chromosomes? Chromosomes from, you see, homologous chromosomes. Bivalent. What is a bivalent? What is a chromosome itself? A full chromosome. What is a chromatid? What is a centromere? And I think as I'm talking about it, you are trying to picture these diagrams or this terminology where you use them. What is a centrosome? What is crossing over? When we talk about crossing over, what do we mean? What is a chiasma? What is a karyotype? What is nondisjunction? What is karyokinesis? And what is cytokinesis? So these are some of the terms that you may come across when you are dealing with the topic of meiosis. Know how to use these terms. Check them. If you are not sure of a term, of which I think by now all of you are familiar with these terms, but there are some terms that you may use interchangeably, or you may confuse them. You may confuse the terms and you think you are talking about a centromere and then you say a centrosome and you mean something else, and then you might lose a mark. So make sure that you use the correct term where you are supposed to. Right. Then we start with the process of meiosis. This process for you, it happens in many stages. So what is happening? Remember we said there are two phases. Phase 1, meiosis 1, meiosis 2. But in meiosis 1 and meiosis 2, there are stages. So let us check what is happening in those phases. If you want to remember them in order, you must come up with a way. So I use this way to remember the stages. So I say I, P, M, A, T. So I only works for meiosis 1. What is I? I is interphase. So if you want to see how I remember that I comes first and then P comes second, I can use this, what is this? I can't remember now, the word can't come what they call it, but I can say Ivan proposed Mary at Thabo. I know you are happy to hear that. So that helps me to remember the order in which the phases occur. Ivan proposed Mary at Thabo. Then what do they stand for? I know stands for interphase. P stands for prophase. M stands for metaphase. A stands for anaphase. Then T stands for telophase. It does not end there. You need to know what is happening in each of these phases. So let's start with the first one, which I said it only occurs in meiosis 1, which is interphase. What is happening during interphase? Interphase is where DNA replication occurs. So you see why you needed to learn DNA replication first. You know that DNA is making copies of itself during interphase. So DNA must replicate to double the genetic material. So each chromosomes will now consist of 2 chromatids joined together by a centromere. So if the genetic material was in singular like this, after DNA replication, because it is going to make a copy of itself, it is going to have something like this. So you have 1 chromatid, another chromatid joined together by a centromere. But in this case, DNA has done what? It has copied itself, or it has doubled itself. In that way, doubling the genetic material. Why do we need to have this genetic material doubled? You might remember, we need this genetic material to be doubled because it needs to be shared equally during cell division. So it helps us to have enough, sufficient, enough. It helps us to have enough genetic material that is going to be shared between the cells during cell division. So remember, we are preparing this cell for cell division. So we want to have enough genetic material that we are going to share between all these cells that are going to be formed. And at the end of meiosis, we know we need to have 4 cells that are formed, which are our 4 gametes. So we need to have enough genetic material that is going to be shared. That is why during interphase, the DNA is doubling itself, or it is copying itself. Now, then there is also a big fight between these two: unreplicated DNA, unreplicated chromosome, and replicated chromosome. In the past, we used to have a question that will ask you at the end of meiosis that how many chromosomes do you find at the end of this process, or how many will you find in a cell at the end of this process? And you will find a cell like this. You will have a cell like this, and then it will have this, this, and this. And using your knowledge, you would say, no, if something is singular like this, we call it a chromatid. But in order for it to be a chromosome, it must be 2 chromatids joined together like here by a centromere. So in your mind, you join this one with this one, and you say it is this one plus this one. You say it's 1. And then this one, you will say it's half then. So you will end up answering to say this cell has 1.5 chromosomes, which is wrong. We don't say that. Remember, if this chromosome appears in a singular strand like this, it means it is unreplicated. So you come here. Unreplicated chromosomes. What is an unreplicated chromosome? It is a single chromosome. So it does not represent a chromatid. If it is just standing there alone, we say it is unreplicated chromosome. It is a single chromosome before it replicates. Then a replicated chromosome is the one. So when you look at this cell again, let me draw the cell again. You will say this one and this one. The correct answer will be there are 3 unreplicated chromosomes. That will be the correct answer. But now, when do chromosomes become replicated? Yeah, this is the story. Once you see a chromosome that has 2 chromatids joined together by a centromere, you must know that that chromosome has replicated. It has doubled itself. Then we can only refer to this thing as a chromatid if it is joined together with something else like this. But if it is just one, you call it an unreplicated chromosome. Please get that. Remember that. Seeing it if you have to. Once it is just standing there alone in a singular form like that, we say it's an unreplicated chromosome. It is not a chromatid. A chromatid does not go alone. A chromatid, we can only call it a chromatid if it is joined together by a centromere with another chromatid, forming a chromosome. forming a replicated chromosome. So a replicated chromosome will be one that has two chromatids joined together by a centromere. Now that one is a replicated chromosome. I think the fight between unreplicated and replicated chromosome is over now. Then here is the story. You see this chromosome that you see here, that the one that is written unreplicated chromosome, it appears in a singular chromosome. It is there. It is just standing there alone. We cannot say this is a chromatid because there is no partner that is joining it to form a chromosome. So it is not a chromatid. Here you see a replicated chromosome. This replicated chromosome consists of two chromatids. This is a chromatid joined together by a centromere. Now, when they are joined together by a centromere like this, only then you can refer to one of them as a chromatid. You see, this is a chromatid joined together, but all of them together, they form a full replicated chromosome. That was just an illustration to explain what we were talking about. Right. So now, how do you identify the phases? There will be diagrams showing you any phase, and they might even ask you what is the name of the phase that came before that one or that comes after that one. So there are key things that you need to look out for, that you are able to identify the phases even without being told what those things are. So how do you identify the phases of meiosis? You have prophase. Prophase is the first one. So prophase, remember we said prophase is the first one, it's the first stage. What is happening during prophase? Follow what is happening during prophase 1. And very important, if you just write prophase, they ask you what is the name of this stage, and then you just write prophase, you are going to get it wrong. Because we are talking about meiosis, there is prophase 1 and prophase 2. When you just say prophase, we take it that you are talking about mitosis. Mitosis, the one that you did in grade 10. So for grade 12, you need to be specific to say, this is prophase 1, this is prophase 2. Any phase that they give you, you must identify at which phase it is. Right. So what is happening at prophase 1? And this is very important because this is also an exam question that they can ask you. What is happening at prophase 1? And also define what is crossing over and also what brings about genetic variation. So there's a lot of things that are happening at prophase 1 that you need to be able to tell that story. Very important phase. So let us take a look. First, they say the spindle starts to form. You see here, this spindle fiber, follow the label there. The spindle fibers starts to form. The spindle fibers are the ones that are going to hold the chromosomes. Remember, you need the spindle fibers. So in a previous cell, they were not there. But at prophase 1, this is where they start forming. 1, you are done with this one. Homologous pairs of chromosomes are visible or bivalent. So homologous chromosomes are the chromosomes from the mother and the father that have the same size, same shape, that are paired together. So you have homologous chromosomes. You start seeing them like this. So once you see them in pairs like this, you know they are of the same shape, same size, same, but from the mother, and one is from the father. So you know that these are homologous chromosomes, right? Right. So crossing over takes place at a point called chiasma. So you start following what is happening here. So there should be some touching and crossing over happening. So once I see this, I know for a fact that this stage is prophase 1, because only at prophase 1, that is where crossing over is taking place. So where does crossing over take place? At a point called the chiasma. Then you come to prophase 2. At prophase 2 is after all these other stages of prophase 1 have happened. But you start again the process at prophase 2. What is happening at prophase 2? The nuclear membrane and the nucleolus disintegrate. Why do you have 2 cells here? 2 cells are coming from telophase 1. Because after telophase 1, you start the process again at prophase 2. So telophase 1 is where now you end up with 2 cells. Now those 2 cells, they continue to further divide. And when they are continuing to divide, they come to prophase 1. So right, at prophase 1, how do you identify this stage? Nuclear membrane and the nucleolus disintegrate. Now the nuclear membrane starts to disintegrate. The nuclear membrane is the one that was enclosing the genetic material. It was enclosing the chromosomes. Now it starts to disappear. You can say disappear here. And then there is no crossing over that is taking place. Because if you take a look at this chromosome here, from prophase 1, it had already exchanged the genetic material. You see this color here is purple. This color here is pinkish. So it means one chromosome has already changed the genetic material, meaning crossing over has already happened at prophase 1. Here, there is no crossing over. That is what is happening at prophase 2. Okay, going back. Another thing that is happening at prophase 1 that you can mention is that the nuclear membrane also here starts to disappear. And the centrioles also start to form and they move into the opposite poles. So this is what is happening at prophase 1. Some of the things that you can add there. Then you come to the next stage. Remember, mat. So you've done I, P, M, which is the third one, metaphase. So what is happening during metaphase? How do you remember this stage? Meta, middle. So you can just say middle. What do you expect? You expect to see the chromosomes arranged in the middle or at the equator in homologous pairs. So they are the pairs. You see, the pairs, they are still there. You don't find them single. So you still find them in pairs to say, this is the pairs from the mother, from the father, from the mother, from the father. So they are still in homologous pairs, but where are they? They are in the middle. So those are the two things that you need to look out for when you are checking metaphase 1. In metaphase 1, we are still talking about homologous in pairs. But in metaphase 2, they are still in the middle. That is how you're going to look for it. Metaphase, middle. They are still in the middle. However, they are arranged singly at the equator. You see now it's just 1-1. Because the first division has already happened. So they were separated during the first division. Now they are arranging themselves later on in the second division. In metaphase, they are arranging themselves at the equator singly. So once you see a diagram that has the diagram like this, it has chromosomes like this. Yeah, I can have them like this. Without any doubt, you must just say, this is metaphase 2. Why do you say it's metaphase 2? Because you can see that the chromosomes are aligned at the equator singly, in a single row. And that is what. And then random arrangement of chromosomes also takes place. So random arrangement of chromosomes. Please do not say random assortment. We no longer allow that term because assortment also means something else. Now we say random arrangement of chromosomes take place. So these chromosomes, they are not staying there in line to say you are for this, you are for this, you are for this. They are arranging themselves there at the equator randomly. And this also brings about variation. So when you asked about variation, you can also mention that. Then the next phase, ma, it's a. A for anaphase. Anaphase means you can remember it by saying apart. Apart. Anaphase, apart. What is happening? Now those chromosomes that were together during the previous stage, what are they going to do? Without even looking at the text, you must see them separating. You must see them moving apart to the opposite poles. Then you must be able to explain what makes them to move to the opposite poles. Then you come to the story. Without even going to the story, just by looking at the diagram, once you see them doing this, and you see that they were homologous and they are doing this, then you know that they are homologous chromosomes moving from the last stage where they were in the middle in homologous pairs. Now those homologous pairs are going to do this apart. But if they're like this, you know it's anaphase 1. But if they're like this, you know it's anaphase 2. So you must just know those without even a shadow of doubt. Okay, let's check what is happening during anaphase 1. Spindle fibers contract. So this is what brings them to pull apart. So cause and effect. The spindle fibers are going to contract. So these are the spindle fibers. Spindle fibers contract. The chromosomes move to opposite poles. Now the chromosomes move to opposite poles. So this is one full chromosome, one full chromosome, one full chromosome, one full chromosome, moving to the opposite poles. Then you must take note. If spindle fibers fail to separate, pair of chromosomes are going to move to the opposite poles. We will talk about this later. So that is called nondisjunction, and this will lead to a gamete that have an extra chromosome, but we know that we don't want that. So anaphase is a very important stage also, because then the chromosomes must separate, and if they do not separate, they bring a problem that we will discuss later. Then during anaphase 2 is the same thing, but the only difference is what? The centromere is going to split. Then the chromatids. Now it's this. Remember, I said anaphase 1. This. They move to the opposite poles like this. Anaphase 2. They move to the opposite poles like this as chromatids. So it means it was one chromosome still splitting further or moving apart. So the chromatids move to the opposite poles. So if you take a look at this one, this is just one single chromatid. But here, it was a full chromosome. So I hope you are noticing the differences between the two. Then the last stage of meiosis is telophase. Telophase 1 and telophase 2. So what is happening at telophase 1? Telophase 1, the nuclear membrane reforms. Remember during prophase we said the nuclear membrane disappear. But now in telophase, it is now reforming. Reforming. The nuclear membrane is reforming, and then the cell membrane constricts. Now this one, the one that is on the outside, is the cell membrane, because it's the membrane that is enclosing the whole cell. It is constricting here, so it is constricting. So to constrict, it does this. So when it constricts like this, so you say the nuclear membrane starts to reform, and then the cell membrane on the outside constricts. And once it constricts, remember you had them at the opposite poles from the previous stage, and then they are going to do this. Once it does this, we call that the splitting of the cytoplasm or cytokinesis. Now this cytokinesis, it is going to leave us with two cells that are formed. And when the cell membrane constricts here, we say it formed a cleavage. So it forms a cleavage like this. This is at telophase 1. That is how you end up with two cells at telophase 1. But because this is not where the process ends, it must still continue to prophase 2. What is happening at prophase 2? Remember I said you start with two cells. These are the cells that you start with. They must further divide these cells in such a way that every gamete that is going to be formed, which are 4, is going to have each the same number of chromosomes. Then they will undergo prophase 2, undergo anaphase, metaphase 2, anaphase 2, then telophase 2. What is happening at telophase 2? Because there were 2. Each daughter cell has half the number of chromosomes. So you check, how many chromosomes did we start with when we started? So we started with 1, 2, 3, 4. So now each cell must have 2. Do you see 2 in each? See, 2, 2, 2, 2. they have been halved. Now, this one and this one, remember I said to you, they are not chromatids. However, they are unreplicated chromosomes because this cell is going to start afresh and replicate itself and so on. Right. Then 4 haploid daughter cells are formed, which are genetically different, and we call them gametes. They are now our sex cells. So this was just to help you identify or be able to identify the stages of meiosis. So what is non-disjunction? We spoke just a little bit about it now, and what are the consequences of this non-disjunction? Now, remember the spindle fibers that we spoke about at anaphase, that they must contract or constrict. Those spindle fibers, sometimes they fail. They fail to separate. So, or they fail to contract and then separate the chromosomes or the chromatids. Then members of one pair of homologous chromosomes fail to become separated. So when they were supposed to go like this, they are like this. They failed to separate when they were supposed to separate like this. So what is happening? During anaphase 1, this was during anaphase 1 when we are talking about homologous chromosomes. Again, if there is non-disjunction of chromosome pair 21, now remember we know a karyotype, we have 23 sets of chromosomes. So when the chromosomes add chromosome 21, remember you have a karyotype 1, 2, 3, 4, 5. You have 23 here, which is very special. And then you have 21 here. Now, if they fail to separate here at chromosome number 21, it leads to an abnormal gamete with an extra copy. So if this gamete is going to fertilize an ovum, it is going to fertilize an ovum with an extra copy of chromosome number 21, then leading to a condition which we call Down syndrome. It leads to Down syndrome. So it forms trisomy 21, leading to a syndrome called Down syndrome. Now, 2 gametes are going to receive an extra copy of the affected chromosomes, and then 2 gametes are going to lack that chromosome. So some of their gametes may not have because it moved. So imagine a gamete that was supposed to be here. It will have nothing because all of them moved into one. So it can happen during anaphase 1 or anaphase 2, right? So we don't need an extra copy at any position of the chromosomes because the chromosomes go in homologous pairs, both for mother and father for a complete organism. Let's say this is a human species, so you need 46. So that 46 is going to be 23 from your mom, 23 from your dad, and then the last one is the one that is determining the sex of that particular organism, if it is a male or a female, if it is a boy or a girl, right? So I hope you understand how non-disjunction works. So you can go back and look at the diagrams that will show you where the chromosomes were supposed to separate and they failed to separate, and now you know what it leads to. You remember that. So this is just an example that shows you what is happening during meiosis 1 or meiosis 2. So the first meiosis division. Let's say you have a normal cell with 2 chromosomes like this. These are homologous pairs. You would expect, okay, let me draw a line here to show you. On this side, these are normal. On this side, these are non-disjunction. These are not normal. So under normal circumstances, let's be here. Under normal circumstances, you would expect this cell to divide and have each copy, one cell have each copy of these chromosomes, 1, 1. That is normal, it's expected. And then when they do the second meiotic division, you expect this cell, therefore the cells that are going to form, to have 1, 1. But in the second meiotic division, you might see that the chromosome here failed to separate and they all came into one cell, and another cell is left with nothing. and this can also lead to a problem. So then at the first meiotic division, if the problem occurred at the first meiotic division, this we would expect it for this to separate and go into this cell and this cell. However, they failed to separate. They both came into one cell. These chromosomes came into one cell. And this will also lead to a process further when they divide under normal. You would expect them to come 1, 1, 1, 1. But what about this cell that did not get anything? It means during the second meiotic division, it will not have anything further. So this is just to explain non-disjunction, what happens to the cells that did not receive a chromosome and those ones that received an extra copy. Then if this happens at chromosome 21, we know it leads to trisomy 21, which leads to, or we call that that situation trisomy 21, which leads to a condition called Down syndrome. Right. So you must also know the differences between mitosis and meiosis. So what are the differences? Mitosis and meiosis. Mitosis, as you did in grade 10, occurs in body cells. Remember, we said these are somatic cells. Then meiosis, like we've just discussed now, it occurs in sex organs. So in your ovaries or in the testes, you must know. Those are the cells that will go a meiosis division, not the ordinary body cells. Then both karyokinesis and cytokinesis occur once. So during mitosis, the splitting of the cytoplasm only occurs once. But during meiosis, karyokinesis and cytokinesis occurs when? Twice, during the first division and the second division. Then at the end of mitosis, you find 2 daughter cells. At the end of meiosis, you find 4 cells are formed, which we call them what? Gametes. The cells that are formed are called gametes. Then the daughter cells are genetically identical to one another and to the parent cell. So during mitosis, everything is the same. Parent, same number of chromosomes. The 2 daughter cells, the same number of chromosomes, same everything. And then during meiosis, the daughter cells are genetically different. Remember, we brought variation and we brought also random arrangement of chromosomes. So it makes the gametes that are formed to be genetically different. And then the chromosome number remains constant. If it was 12, it will remain 12, 12. But here, the chromosome number is halved. And I told you the reason why it needs to be halved, because we don't want to have an organism with more chromosomes. So we need to keep them haploid. So that is the reason what we can. Haploid, or for human beings, you can say 23. Or you can say haploid is represented by N. So these are the differences between mitosis and meiosis. Then we're going to come to the last topic that we need to do for your paper 2. Let us take 2 minutes again just for you to absorb everything that we have discussed under meiosis, and then we'll come back and start with a new topic of genetics and inheritance for your paper 2. Right. Welcome back. Then we continue with the last topic that we'll be doing for today, which is genetics and inheritance. This is quite a big topic. Please make sure that you look at the pointers that I'm going to give to you now. When you go study, then you don't make those mistakes. So this topic, you need to know terminology, monohybrid crosses, and sex determination, sex-linked inheritances, dihybrid crosses, blood groups, genetic lineages and pedigree diagrams, genetic engineering, paternity testing, genetic links, and Mendel's principle of inheritance. So I did not say those in order, but these are the things that this topic comprises of. So let's just get right into it. So here there's also terminology. However, there's a lot of terminology that you confuse when you are writing. Check when you are writing, when you are asked about the chromatin versus chromosome. Look at which one is which. The genes and the alleles. Then you know genes are the segment and then the alleles are alternative forms of a gene. Dominant allele is the one that is going to appear in the phenotype of an offspring, or the one that is dominant over the other. The recessive one is the one that is masked by the dominant one. Then a phenotype is the physical appearance of an offspring. A genotype is the genetic makeup of that offspring. And then homologous pairs, it means they are the same. Heterozygous, they are different alleles. Yeah, homozygous alleles are the ones that are the same. You can say TT, they are homozygous, but T and small t, they are heterozygous. They are different forms of these alleles. So please make sure that you know how to use those terms and you don't confuse them. And then go and revise Mendel's principle or the laws of inheritance. How do organisms inherit characteristics? And then this, very important, you are going to get 3 free marks if you follow what I'm going to say to you. Even if you don't understand what the question was, make sure that you know how to write the format of a genetic cross. So this is how you do it. A genetic cross, the visible trait is the phenotype, example, tallness, shortness, then you must know. Your question is going to have the phenotype because they will tell you if a mother who is this and this, or if you cross a flower that is pink with a flower that is white. So that description that is telling you what you see with your physical eye is your phenotype. So what you must do, you must write phenotype and write that statement or that description that you got from the question. White flower, cross it with a pink flower. You get a mark for that. And that is for free. You got it from the question. And then the genetic makeup of an individual is the genotype. Then you are going to write the genotype. How do you know the genotype? Sometimes some questions will tell you using the letter B, write the phenotypes of these organisms. Then you will check. Did they say the flower was homozygous for pink or heterozygous for pink or homozygous for what? Then you know if it is homozygous, the letters must be the same. Homozygous dominant, the letters must be the same and they must be big. Homozygous recessive, letters must be the same but they must be small. If they say it's heterozygous, you must write letters must be big and small. So that information you are going to get where? From the statement of your question. You write it here, you bring it here, you get another mark for that. And then you shall have written your P1. Whatever that you've just written now, you were dealing with your parents. So your parents are your P1 generation. And then you will break down and separate the gametes. That is after meiosis, because we just said meiosis separates them. So you will write them inside the circles like this and not put a cross there, or you can just put a colon, or even if you put a cross, let them show that they have been separated. And then after that, you write, this is meiosis. Meiosis is going to give me my gametes. Remember, meiosis forms gametes. Meiosis is going to give me my gametes. And what do you do with those gametes? You do fertilization, meaning you cross them. So if you've written meiosis and fertilization, you are going to get 1 mark. And then you've already gotten 1 mark for this, 1 mark for that, 1 mark for writing meiosis at the right place, fertilization at the right place. And please do not write fusion. Write fertilization. Right. And then after that, you are going to get the genotype. Now you're going to get the genes of the offspring. Even if they are incorrect, it's fine. If they are correct, you will get the mark. Then you will give us the phenotypes of the offspring. How many are white, how many are pink, you will also get a mark. Then you will tell us, this now is my first offspring, F1. For writing P1 and F1 at the correct place, we do this, you get 1 mark. So there are actually 4 free marks that you can get, even if your crossing here is incorrect and even if your genotypes are incorrect. A genetic cross is always out of 6 marks. So check if they asked for percentage. If they ask for a ratio, then that may sometimes be a compulsory mark, then you will also get a mark for that one. So make sure that you don't lose marks on a genetic cross or writing that format. Just writing that format, you get marks for that for free. Then, yes, so this is what I was just talking about. Then please remember, go do examples, 3 examples of these. Monohybrid courses, do 3 examples: complete dominance, incomplete dominance, co-dominance, sex-linked, and dihybrid courses. And I would like to emphasize this one. Sex-linked, you have hemophilia and color blindness. Please do not use the X's for everything and say, maybe let's say you use the X. No, read the question and check which one is asking you for sex-linked, which one is asking for a normal cross. So normal cross, you will just cross just the alphabets. And then for a sex-linked, that is where you use the X and the Y only. So this is an example of a complete dominance. That is where the dominant one is going to mask the recessive one. So all of the offspring are going to be like the dominant one. And then incomplete dominance is whereby none of the offspring take the color of their parents. So you see here they cross the pink flower together, a red flower together with a white flower, and then you get pink, but none of the parents are pink. So this is an intermediate one. Both of them are not shown in the offspring. The offspring shows its own color. So co-dominance, you cross a white cow with a brown cow or with a red cow. Now the offspring is going to be roan. Roan is the mixture of red and white. So you have the colors being intermediate. Both of them are equally expressed in the phenotype of the offspring. So you see them physically in the offspring. That is called dominance. And then sex determination, we know that all of these chromosomes are ordinary chromosomes. And then you find these chromosomes there, number 23, which we call them gonosomes. Now, these gonosomes are the one that determines the sex. If you see the big one and the small one, you know it's XY. And this means that this is a karyotype of a male. This is a male. And then you come, this karyotype shows all these are ordinary chromosomes, but this one is that one of a female. Why do we say it's female? Because females have XX chromosome. So when you cross, you remember your female is XX, your male is XY. Then you know every time when a male and a female meet, there is always a 50% chance of getting a boy or a girl. So it's 50-50 each time. Right. Then the sex-linked disorders, remember we just said that you use the X and the Y. Please also follow the question to say, where do they say this disease is attached? Is it attached to the X chromosome or to the Y chromosome? Then you will be able to do your cross there. And then another thing here, you can say a father, if a disease is attached to the X chromosome or to the Y chromosome, the father will only pass that to the boy. But if it is attached to the X chromosome, he will only be able to pass it on to his daughters, not the sons, because to the sons he shall have given them the Y, But the mother can be a carrier, and she can also pass it on to the boy child or to the girl child, or another girl child can also be a carrier of that particular disease. So I think you follow that. And then another disease that you have is color blindness. You know, some people don't see the red and the yellow. You look at this diagram here. Most people, especially males, will say all of these are yellow. Those with color blindness, all of these will look, they don't have the special receptors that will see this as red, this as yellow, this and green. This is called the yellow-green color blindness. So please go check on the blood groups also. When do you use the alphabet? You only use the alphabet for the blood groups, for the alleles. So check how do you use your alleles, the genetic lineages, your pedigree diagrams. Please remember, follow the steps of how to solve a pedigree diagram. Start from bottom upwards and follow the key. Very important. And then you must also know how do you use blood groups to show paternity. There are only 4 blood groups: A, B, AB, and O. So you can only have only those. But this is not a true test to say you are the father, because many people can have blood group A. So this is not a decisive test that we can use. However, you can use a DNA profile because it uses those fragments of the sequence of the nucleotide to tell us that this is the DNA of this particular person, and these 2 people can never share a DNA. Then we can definitely use a profile to test if a person is the father of a particular person. But for blood groups, it is not conclusive. And please go look at the question papers. Gather all the question papers that deals with the topics that we have done today and answer those questions. Test yourself, and good luck for your test on Monday. And thank you so much for watching today's lesson.